Welcome to the VSL Test Interpretation Pod Module Series. This module, Laboratory Pasteurization Count, is brought to you by the Cornell University Milk Quality Improvement Program and the Cornell Dairy Extension Group. In this module, we will discuss why laboratory pasteurization count results are important, background on thermoderic bacteria and the laboratory pasteurization count, how laboratory pasteurization count is performed, and how to interpret results. Laboratory pasteurization count, or LPC, is a test that enumerates heat-resistant bacteria, also known as thermoderic bacteria in raw milk. These bacteria are capable of surviving a heat treatment of 145 degrees Fahrenheit for 30 minutes. When LPC results are high, often considered more than 2 to 300 colony-forming units per milliliter, it may be an indication that there is a cleaning and sanitation or hygiene issue at the farm. The primary raw milk bacterial contaminants that are selected for by the LPC test are gram-positive cocci such as Micrococcus, Staphylococcus, and Streptococcus. Although these bacteria can survive the LPC heat treatment, which is designed to mimic vat pasteurization, the vast majority of these contaminants do not grow at refrigeration temperatures and therefore do not influence pasteurized fluid milk quality. The exception is that a small proportion of the population selected for by the LPC test are gram-positive spore-forming bacteria, which survive both vat and high-temperature short-time pasteurization, some of which can grow at refrigeration temperatures. However, because these so-called cycrotolerant spore-forming bacteria represent a small proportion of the total population selected for by the LPC test, this test does not correlate to pasteurized product quality. Samples of raw milk received through the VSL program are heated in a water bath to 145 degrees C and held at that temperature for 30 minutes. When the heat treatment is complete, samples are rapidly cooled, then plated on standard plate count agar. Agar plates are incubated for 48 hours at 90 degrees Fahrenheit to allow for growth of the bacteria. Following this incubation, the resultant bacterial colonies are enumerated and results are reported in colony-forming units per milliliter. Here is an example of how LPC results are reported on the VSL report form. In this example, the raw milk LPC result of 4,200 CFU per mil is well above the recommended limit of 2 to 300 CFU per mil, warranting further follow-up by the processor. Thank you for watching this module. If you're interested in taking classes with us, learning more about our research, or participating in our voluntary shelf life program, please refer to the information on this slide.